Good evening, Mr. Milamo TK with the YouTube channel Smart_Traveler Memos welcomes you to Biology Practical Marking Tips 2009 Paper 3, which was taking 1 hour and 3/4 hours. 1 and 3/4 hours. That is 1 3/4 of an hour. So, welcome guys without much ado. So, the marking tips. But before we do that is this, ask yourself these questions. Ever wondered what goes on behind the scenes when examiners mark biology practical? Let's dive into the 2009 paper and uncover the secrets that could boost your exam skills. By the end of this video, you'll have a clear understanding of the marking process and the examiner's mindset. You will not only gain practical tips that could transform the way you approach and answer biology practical questions, but we will be setting you on the path to higher scores and greater confidence in your exam preparations. Question 1. The photographs labeled K, L, M, N, and P below are bones obtained from a mammal. For each of the bones K, L, and M, 2 views are shown. So, bone K, view 1, view 2, bone L, view 1, view 2, bone M, view 1, view 2. Then we have bone N and bone P. Okay? So it is only KL and M where you have 2 views, but the bone N and bone P are having only 1 view. But below there, you notice that bone P has got labels 5 and 6. Bone M, view 1 as number 3. Bone M, view 2 as number 4. Bone L, view 2, number 2. And bone K, view 1, label 1, which is labeled with the 2 lines. So we have 1 label in bone K, bone K view 1, 1 label in bone L view 2, 2 labels in bone M view 1 and view 2, and then we have the last labels are on bone P, which is 5 and 6. So we have 1, 2, 3, 4, 5, 6 labels. Bone N is not labeled. Identify the bones and name the part of the mammalian body from which each was obtained. You get 5 marks. So bone K, identity is humerus, found on the forerib. And you have alternative there, front leg, arm, or upper forerib. Reject hand. Where found, you don't say hand, or forearm, or pectoral guido. L is scapula, or you can call it shoulder blade, and it is found on the shoulder or pectoral region. But reject if somebody writes pectoral guido. Identify the bones and name the part of the mammalian body from which each was obtained. We continue. Bone M is femur, or thigh bone, where found hind leg, or hind limb, or thigh, or upper hind limb, or upper part of the leg, or upper hind leg, or thigh region. All those ones there could fit on where it was found. N is tibia, or shin bone, and it is found in the hind leg, hind rib, or leg, lower hind rib, lower hind leg, or lower leg. P is ulna radius. You can see there is hyphen, ulna hyphen radius, which must always be there because it's not two bones, it's one bone. So you have, it is found on forearm, forerib, arm, lower arm, foreleg, front leg, lower forerib. Except ulna and radius if the hyphen is not put. So instead of ulna hyphen radius, you just say ulna and radius. Or ulna or radius, or ulna radius. Reject ulna or radius, and you also reject if one bone is given. All right. So up there you have ulna and radius, or ulna comma radius, or ulna radius. So never accept one bone. Mark independently where found even if identity is wrong or a space has been left. So the place where the bone was found, a student can know but forget the name. So you don't tie. You mark independently. Okay. Name the parts labeled 1, 2, 3, 4, 5, 6. Number 1 is known as condyles. Reject if S is omitted. Or you can say, and you reject lateral condyle, and you reject medial condyle. 2, glenoid cavity. 3, head. Accept hand of femur. Reject hand of humerus. 4, patella groove. Reject groove alone. Patella alone. It is going to be an omission because it is patella groove. So omission means you have left one word. 5, ulna, or shaft of ulna. Name the bones that form a joint with bone K at its anterior end, and name the type of joint they formed. Scapula, or shoulder blade. Reject wrong spelling of those words. Accept spelling error if no meaning, but underline but mark joint. The joint is called ball and socket, but you reject ball hyphen socket, or ball stroke socket, or socket and ball. All those are rejected. Roman 1 is tied to Roman 2. Posterior head, ulna and radius. Reject radius alone or ulna alone. Or radial. Joint is hinge. Accept correct name of joint if radius and ulna, but underline 1 and 2, Roman 1 and 2 are tied. State the function of the structure labeled 6 in bone. P, large surface area for muscle attachment or for tendon or ligament attachment. That's the first function. The other one is that it limits the movement of radius and ulna, or limits the movement of the joint, acts as a stopper or prevents overstretching. 2 points are found there, but mark any and indicate the other as seen. I want first to correct function and see the others. Accept muscle tendon. Reject tendon muscles. Okay? Attachment of muscle tendon, but reject attachment of tendon muscles. Question 2. You are provided with the substances P, Q, X, Y, and Z. P and Q are food substances, while X is dilute hydrochloric acid. Y is dilute sodium hydrogen carbonate, and Z is Benedict's solution. Carry out tests to determine the food substances in the P and Q. Food tests. For that question, for you to mark, you read the procedure carefully when letters are used for food substance and reagents. Accept formula if correct hydrochloric acid or HCl small letters, but reject HCl and HCl. Okay? So you accept the formula if correct. Mark P and Q independently. Mistakes of P should not be transferred to Q. So there we are. I don't know whether we are struggling, but we will try our best. But I think I can see it's quite visible. So we have substance, food substance being tested, procedure, observations, and conclusion. P, note, no need to test for non-reducing sugar because it was confirmed that there are reducing sugars. So, reducing sugar to 1 ml of P and 1 ml of Benedict solution, place in hot or warm water bath or heat to boil. Changes color from blue, green to yellow to orange, stroke brown. Accept green to orange, direct. You heat and then it turns green, orange. Green to yellow, accept. Reject up to green, because that is wrong under. Reducing sugar present. Q, for the food substance, is equivalent mark, because we are testing for reducing sugar. Okay? So, to 1 ml of Q and 1 ml of Benedict solution, place in hot water bath, heat and boil, or heat to boil. No color change or blue color of Benedict's solution remains, reducing sugar absent. Q, non-reducing sugar. To 1 ml of Q and 3 drops of dilute hydrochloric acid, boil for 5 minutes, cool, and sodium hydrogen carbonate till fizzing stops, Add Benedict solution, place in hot water bath, heat to boil. The color changes green to yellow and eventually orange or brown. Reject, no observable change or no change. So conclusion is non-reducing sugar present, or non-reducing sugar has been hydrolyzed, or reducing sugars present after hydrolysis, or but reject reducing sugars present unless it is qualified. All right. So note the procedure here is very, very important, and the order of the way you add the reagents. Because if you have not mastered how to test for non-reducing sugar, you'll be shocked, because if you miss the order, you miss all those marks, and there are many. Okay? 3 drops of dilute hydrochloric acid. Boil, you get marks for 5 minutes. Cool, a mark. And sodium carbonate, a mark. Till fizzing stops. And Benedict solution, place in hot water bath. That is equivalent because it was given up there. Question 3. The photographs WXY are designed show seedlings that were grown under different conditions. Examine them. So WX, they are the ones which are in red pots, and Y and Z are in the plastic containers below. Label 3 parts of the seedling in photograph W, 3 marks. So we have the shoot there. You can see that bracket. So you could label, put that bracket and label shoot. Then we have the leaf, we have the epicotyl, we have the cotyledon, and we have the hypocotyl, which is equivalent mark, because you have already marked epicotyl. And this W, actually, these are beans. Label 3 parts of the seedling in photograph W. This is the way you are marking. Any first 3 labeled parts clockwise, no on 1 or more seedlings. Any first 3 labeled parts clockwise on 1 or more seedlings, except names of 3 parts on the lines provided, only when accompanied by a key. For key mark seen labels, leaf, first foliage leaf, single label line, leaves, 2 label lines. So if you write a leaf, 1 single line. If you write leaves, 2 label lines. Epicotyl, stem point up above cotyledon. The stem point above cotyledon, that is the epicotyl. Hypocotyl is the point below the cotyledon. Then we have cotyledon, node where the cotyledons arise, internode with a bracket from the cotyledon to the point of attachment with the leaves, and then shoot, bracket from base to the top of the seedlings for you to be given a mark. Name the type of germination exhibited by the seedlings and give a reason for your answer in B1 above. The germination is epigyle because cotyledons are above the ground or above the soil. B2 is tied to B1. Seedlings in photograph W and X were planted at the same time. State the conditions under which the seedlings were grown. 2 marks. Seedling in photograph W was grown in the dark, or it was grown in absence of light, or it was grown in insufficient light. Reject dark stage or light stage. Seedlings in photograph X were grown in the light, or they were grown in sufficient light, or they were grown in adequate light, or limited light. When plants are grown in the condition named for seedlings in photograph W, they exhibit a certain phenomenon, and the phenomenon is known as aeration, which is pale color caused by lack of chlorophyll. That's just to confirm, but you just say aeration. State the significance of the phenomenon, or state the significance of aeration for faster growth, to reach light, to obtain light, to search for light, to look for light, to seek light. Faster growth, or once to that effect. D2 tied to D1. Using observable features only, state the three differences between the seedlings in photograph W and X, 3 marks. Long internodes, short internodes. Long stem, short stem. Tall plant, short plant. Small leaves, large leaves, or big leaves. So you can say small leaves, on the other side large leaves, or you say small leaves, and on the other side you say big leaves. Thin stem, thick stem. Thin plant, thick plant. Thin seedling, thick seedling. Yellow, green leaves. Yellow or light green leaves goes with green leaves. Yellow cotyledon, green cotyledon or stem. Seedlings, yellow seedlings, green seedling, and white stem. Okay, we'll go with green. Yeah, we'll go with yellow stem. Okay, green seedling. White stem goes with green stem. Fine. For the table, tick between the differences. The table of the differences. For statement, mark at the end. So if people have drawn a table, you tick at the middle. If they have just made one statement that this plant is tall while the other one is short, you mark at the end. Marking points are 4. So we are just looking at the color of any part, height, size of leaves, thickness of stem. Those are the things we are concentrating on, or which the students should pay attention to. Seedlings in Y and Z were planted at the same time, but under different conditions. Explain how the response exhibited by the seedlings in Z occurred. Seedlings are subjected to unilateral or unidirectional source of light, which causes auxins to migrate or auxins to diffuse to the dark side of the shoot, or more auxin or higher concentration on dark side. All those ones. You see the strokes, please don't repeat the stroke. Causing faster growth, or cell irrigation, or cell enlargement, or cell growth. Faster growth on dark side is tied to high auxin concentration. Accept manipulation of the terms. Okay? Just try to understand what the candidate wanted to bring out. If the candidate brings out that message, mark. Give full marks for higher concentration of axines on dark side. You give 2 marks. Or causing faster growth on that side than the light side, hence bedding. Okay, so that brings us to the end of 2009 marking tips from Biology KCSE Practical, which was paper 3. Expect more good marking tips from Mr. Medamo TK, and please continue subscribing and supporting me after watching my channels up to the end. Otherwise, the next paper will be 2010 paper 3. Welcome, guys.